specific process, I mean what? Their ability to move towards chemoattractant, to sense, stimuli. And as example for that, we walk to the cafeteria to get food. So in environment, the bacteria will walk or move in order to get nutrients, okay? They also move in a way or interact to grow and do reproduction. So all the structure in the bacteria is helping towards doing certain process. Okay, this includes as I said, like responding to stimuli, the metabolism, and growth and reproduction. The difference between the bacterial cell structure, it's very important to be aware of. Why? When I did the introduction lecture, I told you there is certain important difference you should be aware when you discuss the bacterial structure. What is the most important that we mentioned in the previous lecture? The cell wall. Okay. So if I would like to look on the image here, okay. Through this lecture, we will discuss all these details, okay? And I'm not good in drawing, so don't have a minute. If I think of like how we discuss those details, so the bacteria have several structures. It's covered by something called cell envelope, okay? So the cell envelope is if this is the cytoplasm, the first layer, so it's like multi-layer. The first one is the cytoplasmic membrane, okay? The second layer is the cell wall. And in certain bacteria, which we will talk about, they have something called an outer membrane. This is the third layer, okay? Some of the bacteria will also have a structure called a capsule, which protects the cell, okay? Inside the cytoplasm, we have the genetic material for the bacteria, or bacteria is eukaryotic. You remember what is the genetic material in the bacteria? In nucleus. We don't have a nucleus. We have other structure like ribosomes. We have extra chromosomal material, and we have inclusion body. So all these collectively, they play a role to maintain the bacteria structure and the ability of bacteria to process like nutrition, get out, waste, and stuff. And we're going to discuss this in details.
Dr. Could you explain this thing again in front of the camera? Because the people online can see the drawing.
Oh.
Yeah.
I forget that completely.
Exactly, no problem.
Okay.
Do you want me to move the board here, or if you want to explain?
I mean, will I have the possibility to draw something on the...
It'll be difficult, I think, with the mouse.
Okay, so guys at home, I'm going to repeat this. So I was just telling your colleague here that the bacteria's structure is built on different layers that we call it cell envelope. So the cytoplasm is covered with cytoplasmic membrane. After the cytoplasmic membrane, we have another layer called cell wall. And following the cell wall, in some bacteria, not all bacteria, we have an outer membrane. And again, in some bacteria, not all, we might have another layer, we call it capsule, okay? Inside the cytoplasm, we have the plasmid, we have the nucleic materials, the chromosome, we'll have something called inclusion body, and we'll discuss details of the function of those, what we mentioned. Okay, so if I talk about the main structure of the bacteria, the cell wall, cell cytoplasm or plasmid membrane, we have the cytoplasm itself. Sorry, I forget something. In the bacteria, between the cytoplasmic membrane and the cell wall, or outer membrane, there is something called periplasmic space. And we'll have these all details when we show, like, on the screen. Okay, other structure is the mesosome, I forget that, capsule, and then we have externally appendix that help the bacteria to attach or to move. Those are flagella, or flagellum as a single belli fimbrium, okay? So let's start one by one. But before we start one by one, I told you there is the major classifications of they will hear me here too. Okay, the major classification of bacteria is rely on the cell wall component to retain the gram stain, true? So in gram-positive bacteria, if you sit on the cell envelope, you will have the cytoplasmic membrane. I cannot function. Okay, so the green, layer here is the plasmic membrane or cytoplasmic membrane. They are similar in the gram-positive and gram-negative. And then we have the next to the cytoplasmic membrane, the blue line here is what? Is the periplasmic space. If you look on the periplasmic space in the gram-positive and gram-negative, what is the difference? Their arrangements, as the layers, they are the same. But what do you see as a difference? Exactly. The starting of periplasmic space in the gram-positive bacteria is the external part of the cytoplasmic membrane and the internal part of the cell wall. So they are, it's located between the cytoplasmic membrane and the cell wall in gram-positive, okay? In gram-negative bacteria, it's located between the external of the cytoplasmic membrane and the internal of bacterial glycan.
Could you move the mouse because people at home cannot see your laser pointer?
Okay, a lot of technical stuff. Okay, all right, I'll go back. Okay, so if you just look here, in the periplasmic space, so this is like a difference. Another difference, if you look on the cell wall, which is called bacterial glycan, what's the difference between gram-positive and gram-negative? The thickness. So in gram-positive, the bacterial glycan is thick, and in the gram-negative, it's a thin layer. Okay, so this is a main difference you need to remember. Okay, so this is just like a structure, how the cell wall is built. And I'm just trying to do color. As I told you, I'm not good, I'm not an artist. Just try to, live with me. All right. Let us talk about the first layer, which is the cell wall. The cell wall, as I just mentioned, is built of or consists of bacterial glycan, that is protein and polysaccharides. And the protein and polysaccharides or bacterial glycan form like a mesh, strong mesh around the cells. How that help the cells? The first thing is maintaining the cell shape. So the function is maintaining the cell shape. You remember when we talked about the macroscopic and microscopic of the bacteria? We said the bacteria could arrange in cocci, bacilli, and spiral. So to maintain the cell shape of the bacteria, okay? The second thing that it resists the osmotic pressure. So it's kind of like control, because if we don't have this, cell wall, there will be like, molecules, water coming in and out. And we'll talk more when we talk about cytoplasmic membrane. All right, the third thing, they play a role in the cell division and cell wall biosynthesis. So it helps in biosynthesis of the cell wall itself. The major important help in differentiating between gram-negative and gram-positive when it comes to gram staining. Okay. The thickness and the constituent of the peptidoglycan play a role in dividing the two major groups of gram-negative and gram-positive. It's protect organisms, so it's kind of like a protection layer because it controls the effectiveness of antibiotic, harsh chemical, and the cell wall present in gram-positive and gram-negative bacteria, but there is exceptional. So certain microorganisms they have a cell wall deficiency. An example for that is mycoplasma. Following the cell wall, let us just go, towards inside. We have what we call the cytoplasmic membrane. And the cytoplasmic membrane, which a layer surrounding the cytoplasm, the jelly-like water. And the cytoplasmic membrane built of, a phospholipid and more than 200 protein. So it's a phospholipid and protein. This is the constituent of the cytoplasmic membrane. Again, there is exceptional. Mycoplasma, considered one of the microorganisms, they lack sterol. Where did we discuss sterol? Do you remember? Eukaryotics. When we talk about difference between eukaryotic and prokaryotic, can you get the AC? It's not working.
It's not like that.
So what is the function of cytoplasm? The cytoplasm control the interaction of the internal environment with the external environment. It helps in mediating certain process. So it regulates the transport of molecules from internal to the outside, outside the plasma membrane. It's also responsible for a respiratory. And if you remember, the major difference between eukaryotic and eukaryotic is what? Is lacking of membrane, bound organelles, mitochondria. Mitochondria responsible in eukaryotic of the respiration. So the major function of respiration is happening on the cytoplasmic membrane. Also, cytoplasmic membrane involved in the biosynthesis of the cell wall. Okay, so what next? Sure. Mycoplasma? Okay, we mentioned mycoplasma twice. We mentioned mycoplasma when we say that bacteria, gram-negative and gram-positive, have a cell wall, both, but there is exceptional. The mycoplasma, example for microorganisms, have a deficiency on the cell wall, so they don't have a cell wall, okay? So that makes it more resistant to antibiotic because the antibiotic work on the bacterial cell wall, okay? The second example we mentioned, when we talk about cytoplasmic membrane, we said that it built of phospholipid and protein, different kind of protein, and lack sterol, and the sterol is present in eukaryotic. The only exceptional on bacteria is mycoplasma. So this is two, places where we mentioned mycoplasma. That's clear? Okay. We talk about the cell wall, the cytoplasmic membrane. So we need to go, a little towards out. We have the periplasmic space. And periplasmic space is different in what? Different in the location when it comes to gram-positive and gram-negative. So if I would like to identify what exactly periplasmic space, I will say periplasmic space is located internal surface of the outer membrane in gram-negative. Okay? because the cell wall in gram-positive, they don't have the outer membrane. So they have the cytoplasmic membrane and the cell wall. The area between in gram-positive, the cytoplasmic membrane and outer membrane, a little tiny. This is the periplasmic space in gram-positive. In gram-negative, it's from the external to cytoplasmic membrane and the internal of, outer membrane. Clear? So the periplasmic space contains what we call peptide-glycan layer and a gel-like solution of protein. The function of the periplasmic space, it's a compartment where the transport system works. So it transports protein, sugar, ion, and any other metabolized. In the periplasmic space, we also have a degradative enzyme, and those enzymes will break down larger molecules, to small pieces. So they work on, breaking down macromolecules. Another function of the function happening in the periplasmic space, they also detoxifying enzyme, which considered to be a virus factor. Example for that, the beta-lactamase, and beta-lactamase act, against the penicillin as antibiotic. So, okay, are we clear? We went out, went toward the internal area. What else? The outer membrane. And outer membrane, remember, it's only where? In gram-negative bacteria. The outer membrane is a symmetric bilayered structure. So what does that mean? When we say it's a symmetric bilayer structure, it's available in gram-negative, external to the cell wall. It's connected to the cytoplasm, membrane at the adhesion site. And when I say it's symmetric bilayer, because it's composed of protein and phospholipids. The protein is embedded in the layer. So this is why we call it a symmetric bilayer. The outer membrane maintains the structure of the gram-negative bacteria and work as a permeability barrier to large molecules. What that means? So it controls the passage or transport of larger molecules. Okay? It's also work as a permeability barrier to control the hydrophobic molecules. An example for these hydrophobic molecules is antimicrobial, like antibiotic. The hydrophobic molecules and the larger molecules can actually pass through certain protein. We call them what? We call them purines. So if you wanted to define what's purines is protein integrated in the outer membrane. Their function is to mediate antibiotic resistance and to control or allow the passage of the small molecules to internal cells. All right, so we kind of like come to the end of the cell wall, or cell envelope. Yes?
What do you mean by mediate antibiotic?
Okay, so I just mentioned on the outer membrane, the outer membrane control. Let me just go back to previous slides. The outer membrane control the hydrophobic molecules. So if you think of like how that happened, it allows the passage of the small molecules, but not the larger molecules. So antibiotic will not be able actually to cross. And the small molecules will pass through, a protein called purines. this is why when you think of gram-negative bacteria, they are more resistant to antibiotic. So, is that clear? Okay. Okay.
Okay.
Okay. So external to. Okay, I don't want to say that. You answer me. So capsule. Where do we have capsule? In gram-negative. Okay. External to the outer membrane in gram-negative bacteria and cell wall in the gram-positive bacteria. So yeah, I mean
Double click. Okay, I have a question. And before you mentioned hydrophilic or hydrophobic, could you specify which one is it? Because the presentation said hydro.
Topic.
Yeah.
Okay, I can just go back to that.
Okay.
All right. So, okay, capsule is an external layer that covers outer membrane in gram-negative or cell wall in gram-positive bacteria. The function of the capsule, we will talk about it in detail, but what is the compositions of the capsule? Capsule is composed of polysaccharides that could be like starch or amino acid, and it's different in certain bacteria. Example is anthrax or Bacillus anthrax, where they have amino acids as the constituent of the capsule layer. In some bacteria, capsule could be arranged neatly, or we call it capsule, but they could also be as diffused, layer, and we call it slime layer. Okay, so if they arrange like neatly, we call them capsule. If they arrange on, diffused structure, we call them slime layer or biofilm. Capsule is not important for cell viability. So if the cell or the bacteria have a layer of capsule surrounded, it's not affecting whether the cell is viable or like dead. When we talk about the function, I can just explain it more. And just remember, not all organisms or microorganisms have capsule. So certain bacteria will have, some bacteria will not have the capsule. So what is the function of the capsule? Yes?
Is the slime layer like more permeable than the capsule? Or what's the difference?
It's the arrangement of the capsule. If you have it as like cortex layer surrounding the membrane, it's called capsule. So it's a cover. The slime layer is more diffused around. I think I have, yeah. If you just look on the image here, so this is how the arrangement of the slime layer around the outer membrane. And if you have it as one, tiny like layers surrounding the outer membrane, it's called capsule. So what's the function of capsule? The function of capsule is to help the bacteria to adhere. That's one. But the most important, capsule considered is a virulence factor for, encapsulated bacteria, because not all bacteria have capsule. It plays a major role as a virulence factor. It helps the cell from, toxic material. It helps the cell in, promote concentration of nutrition, keep like nutrient inside. It plays a role in the adherence. That means colonization. You remember when we talk about the major virulence factor or the ability of pathogen to cause a disease, the pathogen need to be, enter, adhere, enter, colonize. So encapsulated bacteria will, be able to adhere and colonize inside the host. It protects bacteria from phagocytosis. So if you have a shell of capsule around the bacteria, it will overcome the immune system because it will not be recognized, so it will not be eaten by phagocytic cells. Okay? It also protects the bacteria to be engulfed or infected by viruses. We call it bacteriophage. And the capsular material is antigenic. So antigenic, that means it could be used as a means of identification. That's another thing. And if I think of example of encapsulated bacteria, you have Klebsiella and you have Streptococcus pneumoniae. So those are examples. All right. Getting back internal to the cell, we talk about all the cell wall layer, including the exceptional of presence of capsule and absence of capsule. Let's talk about the internal structure, the cytoplasm. The cytoplasm is made of water, so 70 to 80% of the cytoplasm is water. And then we have amino acid, we have the different components we talk about in that area, like the ribosomes, the plasmid, and the nucleoid or the DNA or genetic material of the bacteria. So they also have something called inclusion body, and inclusion body, there is disagreement, in the role of this tiny inclusion body, and that's part of the research. So don't worry about it. All right. The cytoplasm is a place where the growth, metabolism, and replications happen. So it's an area where all these functions actually performs. Okay. As I said, there is a tiny bodies here. We call them inclusion body. Inclusion bodies are responsible to store energy. They could be available in different sizes inside the bacteria, and they use when the environment is harsh on the bacteria. So when the nutrients is depleted, the inclusion body could be the place where help to supply. Okay, and researchers say that inclusion body could aid in identifications of bacteria, but as I said, recently, there is disagreement on that, statement. All right. Inside the cytoplasm, we also have mesosomes. So what is the mesosomes? Mesosomes is, structure coming from the infold of the cytoplasmic membrane. So you have this, if you look on the image down the slides here, you see the cytoplasmic membrane, it's infold towards inside. So this is what mesosomes. Okay? The mesosome could be, available in different structure. If you see the image on the top, there could be vascular, there could be tubulars or convoluted or multinucleated. Okay, the structure of this mesosome, as I said, it's formed as result of infolding of the cytoplasmic membrane or plasma membrane. Just remember, cytoplasmic membrane and plasma membrane are the same. Okay. So the mesosomes are more predominant or available in gram-positive bacteria, and they play a role in the respirations of the cell compared to mitochondria. Can I compare them to mitochondria? No, they're not comparable to mitochondria. Although they play a role in respirations, and mitochondria are responsible for respirations in the eukaryotic. Why I cannot compare them to anaerobic? No.
Because it doesn't have a layer.
You're close to answer because the mitochondria is an organelle, so it have the complete feature, have a nucleus and stuff. Here, this is as a result of enfolding of the cytoplasmic membrane to increase, enlarging the surface in order to perform the respiration. All right. Nucleic material like DNA. All right. What are the genetic material in the bacteria or in eukaryotic? Is what? The chromosome, plasmid, singular circular DNA, which we call extra chromosomal, material. All right, great. So the chromosomes is the single, chromosomes or located freely in the cytoplasm, and this is why we said this is the major difference between the eukaryote and eukaryote. I'm so sorry. So it's freely in the cytoplasm. They don't have any, membrane around. It's, as your colleague was saying, it's circular, double-strand DNA. In some bacteria might be linear. Naked DNA mean what? Mean they don't have membrane bound, so they are freely in the cytoplasm, and they are actually condensed distantly in an area called nucleus. So this one we already mentioned. The plasmid is considered as an extra chromosomal, DNA, sometimes integrated in the chromosome and contain gene for replication. So plasmid, besides they contain gene for replication, they play a major role in antibiotic, resistance. Why? Because they contain gene, resist to certain antibiotics. This is one of the most important functions of that. Okay. When we think of, we will discuss in bacterial genetics, we'll talk about, the plasmid and the role in providing, bacteria, the ability or, selectivity for certain antibiotic against, other ones. So this is why the plasmid play a role in the bacteria genetic, especially in something called conjugations. I don't want to go toward details of that, but just recall or put your notes, and we will talk more when we talk about bacterial genetic on that. Okay. Other than the chromosomes and the plasmid, we have the ribosomes, and ribosomes is rounded granules floating on the cytoplasm or in cytoplasm. They are actually present in both eukaryotics and eukaryotics, so they are in bacteria and eukaryotic cells. They make up the nucleic acid and the protein. So their function is to make the nucleic acid and the protein. So they consist of a small, 70 subunits. So it's small 70 subunits, that's 50S and 30S subunits. You remember the eukaryotes have what? A larger, which 80 divided into or build up of 40 and no, big no. 40 and 50. والله يا شباب عيب الكلام. Okay, I'm answering this question. Go to the table. All right. The appendage or external to the cell wall or the cell envelope, we have a structure help the bacteria to move, that's flagella, or villi as helping the bacteria to adhere, but also play a major role in the genetic exchange or the exchange of genetic information. All right. The flagella is long filament this appendix, on the surface of the, bacteria, help or providing swimming movement, enable bacteria, to move or to be motile. The flagella is built of a protein called flagellin. So the flagella built from flagellin protein, which have, antigen H. Antigen H could be as, a source of identifications. So you can identify the antigen H, but also could be target for, the protective immune response on bacteria have flagella. The flagella could be divided into different groups based on the positions of the flagella on the surface of the, bacteria cell envelope. The first one is when the flagella arrange as a single flagellum at one end, pole. We call it monotrichous. Okay? So if the flagella presents as a single flagellum at one pole, we call it what? Remember the spelling, monotrichous. Okay. If the flagella is single, but at the two sides, or both poles, we call it amphotrichous. We have arrangement as two or more at the end of both poles. We call it. If you have them arranged as two or more than two at one end of the ball, we call it lophotrichous. And when the flagella is surrounded the cell wall all over, we call them vitritrichous. Okay? If you look on a bacteria blade in the lab and you see like swarming feature, that means these pathogens are very motile and they have, the flagella. Another organelle structure is the fimbria. But let us talk about the exceptional. So how the flagella move? The flagella move by motor apparatus. And if you see here, you have the flagella, which is, external to the outer membrane in the cell wall of gram-negative bacteria, and external to the cell wall in gram-positive bacteria. The flagella connected to a rod, and this rod is connected to what we call a disc or basal body, and this basal body is controlling the, rotations of or movement of the flagella. Okay? So this is the summary of what we discussed. If I think of how what's the exceptional, some species of bacteria, they have a different type of flagella. We call them endoflagella. Okay? So what's the difference between the flagella and endoflagella is the positions of the flagella. The endoflagella is actually moved in the cell through, torsion exert by endoflagellar rotation. I'm going to show you a video. It's a bundle of exofilament, and if you want to see how it's moved, so you see the external is the flagellum, you go connected to the rod, and then this is the disc. The disc could be like 1, 2, or more. Okay. The last, I think this is the last. So the last, structure that I would like to discuss is villi or pilus as a single or Fimbria. So they are present in gram-negative and gram-positive. The Fimbria present in gram-negative and gram-positive. The pili is, more in gram-negative bacteria. Okay. They are short, thin structure attached to the, external of the bacterial cell wall. They responsible for facilitating the attachment of the bacteria to the host. And of course, could be also, a means of helping the bacteria pathogenicity because it helps the bacteria to attach. So this is, one of the steps we just discussed when we talk about how the pathogen could be successful to cause a disease. Okay, the pili is shorter and stiffer than flagella. So if you see the flagella, which have like long, filaments, this is the difference. They are more like, shorter, and they build off a protein called glycine or fimbrine in case of the Fimbria. They are divided into two types, and the division here is connected to their, role. So we have the sex pili, which mediate DNA transfer between, mating bacteria. If you have like the bacteria meeting together, the pili will connect the bacteria and allow I think I have an image. It will actually facilitate the DNA transfer. And we will discuss that in details when we talk about bacteria genetic and talk about a mechanism called, conjugations. So here, this is like, a donor cell, this is a recipient cell, and then the pili is connecting the two cells in order to transfer DNA, material. Okay, the other one is the common pili, and this is like, help bacteria to adhere to the host cells and also protect the bacteria from being eaten by the immune cells. So it protects bacteria from phagocytosis involvement, and this is considered, as a virulence factor. All right. We just talk about Fimbria. It's also help in the adherence of the bacteria to the host, and the other thing, the adhesive properties of the Fimbria actually will actually play a role in the pathogenicity. Again, the ability of the pathogen to cause a disease, they need to adhere, colonize, and multiply. It's also immunogenic, and it have something called antigenic variations. Antigenic variations when bacteria will be able to have different antigens, so it will not be actually detected because they have variables, antigens like changing. All right, so this is my last slides. Bacterial structure and function are 3 lectures. We talk about more general information. So if I just would like to recap on the what we discussed, okay? So we talk about bacterial cell wall. We talk about bacterial cell wall, bacteria envelope. And in bacteria envelope, we talk about different layers and different, functions for those layers. We talk about starting from internal, we have the cytoplasm. External to the cytoplasm, we have the cytoplasmic membrane or plasma membrane, and we have the cell wall after. The cell wall is maintaining the bacteria shape, okay? In the cytoplasm, we have the genetic material, that's the chromosome, and the plasmid and the ribosomes. External to the cell wall, in some bacteria, we have an outer membrane, okay? An outer membrane is considered protecting gram-negative from antibiotic. So this is why we have, because it's control the passage of certain molecules. This is why gram-negative are more resistant to antibiotic compared to gram-positive. When we talk about the cell wall, we talk about the thin cell wall that's built of peptidoglycan in gram-positive, while thick in gram-negative, and we mentioned that it's play role in identifications gram-positive or differentiate, between gram-negative and gram-positive. So this is the measure, function or difference between gram-negative and gram-positive, the constituent of the cell wall that contain the gram stain. We talk about certain bacteria will have a layer that protective called capsule or slime layer, depends on the arrangements. And we talked about external appendages, flagella, villi, and fimbriae that help the bacteria in movement, adherence, and they are considered to be part of the variance factor of the bacteria. Okay, take home message, sit down, draw an image, and try to know the position of these structures, their function to maintain the reproductions and growth of the bacteria. Okay? That's all. You guys have question to me?
Yes.
Yes, I know there is a question from people at home.
You want me to ask them?
Yes,
Okay. So, Doctor, we have 4 questions. One of them, the first one is that they're talking about the foreign channels. They're saying the foreign channels, do they transport hydrophilic or hydrophobic molecules?
Okay, let me just open my slides here. And outer membrane too. So borings will actually permit the passage of low molecular, weight hydrophobic molecules.
All right, Doctor. The next one is she's asking how do we identify the flagella? Do we identify them based on whether they have the H antigens present or not?
Oh well, the thing I mentioned, I said because flagella is built of protein, and it's very antigenic because they have something called H antigen, and this antigen could actually be a source or means of identification. But also, when you talk about how to differentiate between different type of flagella, you have to rely also on the presence of or positions of the flagella on the wall of the cells.
All right. And speaking of flagella, there's one more question. She's asking, could you please re-explain the movements of the endoflagella and the external flagella?
Okay. So if I talk of the eukaryotic flagella, I'm talking about the regular flagella that rotate by motor apparatus, and this motor apparatus is you have the flagella external to the surface, and then you have connected to a rod, and this rod connected to a basal body, we call it a disc. This disc could be like 1 or 2 or or more. And the difference between the flagella and endoflagella is the I don't know if I missed that, but the endoflagella present a special type of bacteria called spirochetes or spirochetes, and the flagella does not actually the difference that the flagella is not actually protrude through outer membrane, and in the endoflagella they actually it's just spread around internal to the cell. So it's not external. They rotate. The rotation will be, as what we call torsion, except by the cell. So it's internal to the cell. So it's just embedded within the cell.
Hi, Doctor. And the last question, she's asking, can you please re-explain the capsule and the biofilm?
Okay. The capsule biofilm. So the first thing that not all bacteria have the structure or the capsule. So the capsule is present in some bacteria, absent in some. The function of the capsule is protective, of bacteria from natural environment. It's built of polysaccharide that could be starch and glycolipids. There is exceptional in the certain bacteria such as anthrax, built of amino acid. So if the capsule arranged neat around the bacteria, we call it capsule. But if it appears as diffuse layer surrounded the cell wall or the outer membrane, we call it slime layer. The function of the capsule, as I said, it's protective, but also play role in the virulence, because it helps the bacteria to to colonize to the host. It helps the bacteria to be overcome the immune response, so it will not be killed or phagocyted by phagocytic cells. And the capsular antigen could be also part of the serological identifications of bacteria. Okay.
Yeah.
More questions? All right. Feel free, if you have more questions, you can email me.
Perfect. Thank you, Doctor. Should I end the meeting now?
Oh, yeah.
Oh,
you took that in there?